Dear students, I am Assistant Professor Sanjeev Kumar Singh, and I'm going to discuss DNA repair mechanisms, which is in the syllabus of BSc third semester Botany, NP University. So before we begin to understand what is DNA repair, we should also have to understand what is DNA damage. First damage happens, then repair happens. DNA gets damaged because of many reasons. It may be endogenous or exogenous. Endogenous factors are some metabolic errors, metabolite errors, or replication errors. Exogenous aspects includes mutagens, environmental factors, radiations, and there are a plethora of those things. Now, when the damage is done, there are different mechanisms, but broadly there are three basic mechanisms through which DNA repair takes place. The first is base excision repair. The second is nucleotide excision repair. And the third is mismatch repair. Now, I'll be dealing separately in different videos. In this video, I'll be dealing with base excision repair. Now, we all know what a base is. So, sometimes DNA bases are modified or they're damaged by the process called deamination, alkylation, or maybe oxidation. Deamination means removal of the amino group. Alkylation means addition of alkyl groups. So these all lead to damage in that nucleotide base, and it leads to wrong pairing during replication, which might result into a mutation, and it will affect the gene or the polypeptide structure. So this modified base position is referred as AP site, a basic site, or you can call it a pyrimidinic site or a purinic site, because the purine or pyrimidine is altered over there. Now, the first step in this base excision repair, it itself means that the base is excised. So the base is excised by the enzyme. The first step is the enzyme glycosylase recognizes this AP site and removes its base. Now, the characteristic feature of this DNA glycosylase is that it removes only the base, but lets the phosphatic or carbohydrate moiety intact. So when now the base is removed, there's a gap. So this gap is filled by not this gap. The AP site, the base is removed. Now another enzyme that is called AP endonuclease. It removes the AP site, that is the purinic or pyrimidinic site, along with some of the neighboring nucleotides adjacent to it. Now this gap is filled by DNA polymerase 1. That is the enzyme which is responsible for synthesis of DNA. And after the strand is synthesized, there is gap from which the site was cut, and this is joined by an enzyme called DNA ligase. So we will now go through the diagrammatic aspect of this whole process to make it more clear. Here is the diagrammatic representation of that. So you can see these two strands of DNA, 5' to 3', 3' to 5', and you have the damaged base marked with red color. Now this DNA glycosylase enzyme removes the base there and creates that AP site. Now this AP site is removed by this AP endonuclease enzyme, and a gap is created here. This gap is now first synthesized by DNA polymerase 1 and when it is synthesized, you can see it in red structure. A gap is left over here. A gap is this portion is left here, this portion is left here. Now these two are joined by the enzyme DNA ligase, and the repair mechanism is complete. So in next video I will be discussing the nucleotide excision pair, and in next video I will discuss the mismatch repair. Thank you.